One example of the many visual deficits correlated with ASD is a condition called nystagmus, which is the involuntary movement of the eyes. And children with ASD are about 5 times more likely than typically developing children to have this condition. So in addition to the broad visual deficits that we see in ASD, a number of studies have also found that specifically retinal function is altered as well. So a previous study has found that retinal function is altered in humans with ASD, particularly Fragile X syndrome. And this study found that the A wave in patients with Fragile X syndrome was not significantly altered compared to the control group. However, when they looked at the B wave, they found that the B wave amplitudes were significantly decreased in patients with Fragile X syndrome, which is suggesting that there is some deficit in the interneurons in the retina of patients with Fragile X syndrome. So I'm really interested in knowing if the circuits that are mediating waves are altered in our ASD models, and the way I'll be doing this is by utilizing pharmacology. And specifically, I'll be using a nicotinic acetylcholine receptor antagonist called DH-beta-E, and this will block the nicotinic acetylcholine receptors on the starburst amacrine cells and retinal ganglion cells. So during the time of cholinergic waves, there will still be acetylcholine, but they will be unable to bind to the receptors and waves will go away. I'm showing you some traces of before I use DH-beta-E and after I use DH-beta-E, and we see that before DH-beta-E, we have these peaks which correlate to wave activity, but then after DH-beta-E, we see that the wave activity is abolished, and this is confirming that the circuit was mediated by acetylcholine. And we see that in wild type, after I've used drug, we see that there is an abolishment of waves. It goes from about 1 wave per minute to no waves. And we also see something very similar in the FMR1 knockout, which is showing that in the FMR1 knockout, they're still being mediated by acetylcholine.